Hey, good morning, everybody. It is Wednesday, and we're on day 3 of this shutdown. Hope you guys are doing good. This is the video for AP Biology, so if you're in freshman bio, you can turn it off, or if you want to learn about the immune system, you can keep it on. Totally up to you. Let's see. Try not to rush through all this, but so this is kind of the first video we've made for AP Bio, and by now you should have watched the body story flu video yesterday. I know it's a little cheesy at times, but it gives you a good visual for what's going on inside your body when you get sick. And now with the coronavirus and everything, again, it really couldn't be more relevant. So hope you got something out of that. Today we're going to go into the second part of the immune system chapter, which is chapter 43B. You guys are working on the reading guide for that tonight, and so I'm going to walk you through all of that, and hopefully it makes sense. We do have an extra resource today, and that is a immune system comic, and you can download this on Canvas. It looks kind of like this. Let me drag this over so you guys can see it. Over here. There we go. Okay, so here's the immune system comic, and the first 3 pages are just like a cartoony overview that actually I drew up for my freshman years and years ago. Actually, it was my first year teaching. We ran out of time, and I drew a comic to keep them entertained. But at the very end, there is a diagram here. I'm not going to give you all the spoilers, but this diagram here is really money. It's got everything you need to know that we're going to go over today, and it'll make more sense when we get to that, but it'd be a good idea to make sure you download that. You could even pull it up as we are going through the PowerPoint today, and that way you can kind of follow along. So let me go ahead and start this present. This is only my second time doing this, so we'll see how this goes. There might be some technical difficulties. Okay, so let me see. Hopefully it started. Yeah, it started where I wanted. Okay, so we are going through the second half of chapter 43. And just a quick recap for you. When you look at your immune system, it's broken up into 2 parts. You have your innate immunity, which is what you're born with. And this includes all the stuff we talked about before we closed school, your external defenses, your internal defenses. And the goal here is just to kind of slow down the movement of the pathogens into your body. And so let's use coronavirus as an example, right? Right now, what you're hearing a lot about with social distancing and good hand hygiene is we're trying to give those external defenses a boost, trying to make sure that any of the viruses that are on our skin are getting washed off with soap, and that we're also trying to avoid those viruses getting into our orifices, our mouths, our nose, and in there we do have mucous membranes, but they can definitely sneak past that. Now, if the virus gets inside of you, I'm okay, just had lunch, so. If they get inside of you, then your first line of defense are your internal defenses. And those are going to be your phagocytic cells. Those are really the big soldiers here, right? We talked about, these are also known as leukocytes, but things like macrophages, dendritic cells, and neutrophils, what they do is they use phagocytosis to engulf any pathogens that they might see, and then they'll use lysosomes and digest them and break it down. This might be one of the reasons why a lot of young people aren't showing symptoms of coronavirus, is just that if it's being taken care of early on, then very few of those symptoms are actually being activated. Now, the problem with these internal defenses is, remember, they are non-specific. So we're going to talk more about this today, but the coronavirus on the outside have specific glycoproteins, right, that are unique to this COVID-19 virus. And these phagocytic cells, they can recognize that, hey, that doesn't belong here, but it's not going to be able to know, like, hey, that's COVID-19, and mount a really specific attack. And that's what we need the acquired immunity for. That's what we're going to look at today, is this really specific response to a specific pathogen. And that's going to involve 2 parts to it. It's a 2-part battle. You have your humoral response and your cell-mediated response. So the way your acquired immunity works is you use these special cells called lymphocytes, and they sound like the same as leukocytes, but you want to keep those two separate. Leukocytes are those dendritic cells, macrophages. Lymphocytes are kind of like your SWAT team, Navy SEALs of your immune system. They are way more specialized. Now, like the leukocytes, they do arise from stem cells in your bone marrow. But depending on where they finish developing, they get their name. So all of them start in the bone marrow, but B cells continue to mature in the bone marrow, and that's why we call them B cells. B is for bone. The other group, they're going to migrate from the bone marrow to the thymus gland, which is sits right here in your neck, just kind of above your lungs a little bit, and they're going to mature there. And so those cells, because they mature in the thymus, are called T cells. T for thymus. And there they are. Now, the way that the acquired immunity works is it comes back to this word antigen, which we talked about before. An antigen is a foreign agent, it's a chemical, right, that can be recognized by your immune system. And the reason it can recognize it is because on any given antigen, where's my mouse? There it is. They have these little proteins called epitopes. And these are also known as antigenic determinants. So you don't need to worry about that. All that means is these are areas of the antigen, of the virus, that can be recognized by our immune system. And the hope here is that when a virus like coronavirus comes in your body, that you are going to have antibodies, you're going to have B cells and T cells that have a binding site that is specific to one of these epitopes. Okay, and that's what actually allows them to be really, really specific in how they attack, is that there's a binding site there. So you got to remember, for cells, our cells don't have ears, they don't have eyes, they don't have brains. If they are going to recognize something and then trigger some kind of cellular response, all of it is really about chemicals. It's all about chemical structures, right? It goes back to something we've been talking about all year long. So these lymphocytes, these B cells and T cells on the outside, they are covered with these specialized receptors. And you'll see more of them in these in pictures that are to come. But they basically look like either just straight kind of bar-like shaped structures, or they can be Y-shaped structures that at the very end have a specific binding site that can recognize one of the many epitopes on an antigen. And when it binds to it, then it'll trigger an immune response. So let's take a closer look at these receptors. B cells have these receptors kind of built into their membrane, and they have thousands of these. Like one B cell or one T cell can have 100,000 of these receptors. And the structure of these receptors is that all of them have this area here. It's called the C region. C stands for common, and then they have a V region, which stands for variant. So all these receptors have the same C region, but the V region can vary. And actually, what a really cool thing is, it all has to do with gene regulation. And so in our immune system, in our DNA, in our genome, we have all these different immune genes. And to kind of tie it in with a previous unit, do you guys remember how we talked about alternative gene splicing, right? How introns and exons get cut out and rearranged to make different versions of a protein? Well, this is where it becomes really valuable for us. And the book has a good diagram. I couldn't find it to put in this PowerPoint, but let me grab the book here. The diagram that we want to look at is on page 906 in your book, figure 43.11. What that shows, I'll kind of show you what it looks like here, like that. What it shows is are two genes, two gene groups. Okay, one they call the V genes, and they're numbered. The little exons are called V1 through V40, and then you have another region called the J region, which is J1 through J5. Right, so you have like V1, V2, V3, V4, and then you have J1, J2, J2, and so on. So when these lymphocytes are being made and developing in your bone marrow and in your thymus, what the cells will do is they will randomly grab one V region and one J region and put them together, right? So you could put like J1 with V1, and that's one version of that variable region on your protein. But then you could have J13 with V10, and that would be another version. So when you mix all those different combinations, you end up with a lot of different kinds of proteins, and there are a lot of different immune genes in our genome. And that's how you're able to make, there's an estimated like million to up to 10 million different kinds of receptors that your body makes. And it's all made by using this alternative gene splicing, by using different chunks of different genes to make different combinations of protein. And so the hope here is that by the time you're born with all these B cells and T cells, right? And as the video said yesterday, you don't have that many of them. You may only have like 2 B cells and 2 T cells for coronavirus, which is why it takes some time for you to get better. But the hope is if you have a B cell or T cell that has a V region that matches with coronavirus, then your body's going to at some point be able to find that match and then mount a response. What's scary is if you have a pathogen that has an antigen or epitopes that can't be recognized. And those are the ones that, no matter how hard your body's going to try, if it can't get that exact match, it's not going to be able to mount a response. Okay, so kind of just an overview of how this works. Actually, I don't really love this part here. We're going to come back to these slides. Actually, you know what? Maybe I'll just talk about it, because I might forget. It's a little different when you're doing this online. So let's go through a couple of things. So let's talk about B cells first. So B cells on the outside have this Y region shape, and they're structurally really similar to antibodies, which I'm going to talk about in a sec. Antibodies are different than the actual receptors. Okay, but here's how you got to remember it for now. If a B cell has a particular variant region that matches, let's say, coronavirus, what the B cell is eventually going to do is produce millions and millions of antibodies, okay, which are basically just millions of proteins that are not mounted in the membrane. These proteins are released into your bloodstream, into your lymphatic vessels, and any virus that's like floating around inside your body, that V region will recognize it and bind to it and basically flag it for destruction and prevent that virus from taking over. Okay. Now with T cells is a little bit different because they also have that region that can bind to the receptor, but they don't make antibodies. What T cells do is they bind directly to antigen fragments. And that's going to be really important because the job of the T cells is actually to attack all the cells that have been infected. Okay. Now we've seen this before with the natural killer cells, right? They were able to locate a cell that had been infected and said, hey, sorry, you're one of us, but you're a risk to all of us. And so the natural killer cell sprayed the chemicals, it triggered apoptosis, and it killed that cell. Well, T cells can do a similar thing, but it's much, much more specific. And so where you're going to see the receptor, this variant region, interacting with infected cells is with this, let me move my face over here, is with these molecules called MHC molecules. And MHC molecules, you can look this up in the book, that stands for major histocompatibility complex. It's a big word. All that means is that these are a set of proteins that have the ability to present an antigen. Okay, and I know that sounds kind of weird. I'm looking for props here because I don't have any real props. Here, I'll use this USB drive. So here's how you can think about this. If this is the virus, right? This USB drive is not a jewel. If this USB drive is a virus or an antigen, it can be presented in a couple of different ways. So number one is, let's say this virus gets into your cell. That virus is using your cell to replicate and make more and more viruses. What your cell has the ability to do is to steal a piece of that virus. Okay, we're going to just call it the antigen. And what it'll do is it'll mount that onto a protein called the MHC protein or MHC molecule, and it'll hold that outside of its cell membrane. So you can see if you look closely here, I don't know if I can zoom in. I can. Cool. Right here, that green protein here, that's the MHC molecule. And what it's done is it's taken this blue antigen fragment, mounted on there, and then it's displaying it on the outside of its cell. Okay? And what that's doing, that infected cell is doing, is basically saying, hey everyone, I'm infected, and not only am I infected, this is what's infecting me, and it's holding out that antigen so that hopefully it can be recognized by a T cell or a B cell. Okay? And that's the job of MHC molecules. Now you'll see here that there are two types of, doesn't like it when I do that. There are two classes of MHCs. And I'd like you to know them. Okay, you have class 1 MHCs, if you want to jot this down, are displayed by cells that are infected. The other MHC, though, is called class 2 MHC molecule. And these are displayed not by cells that are infected, but by cells that have already come in contact with the antigen. Now, what cells have already come in contact with the antigen? Well, keep in mind that by the time we get to that late stage, that acquired the active immunity part, all your innate immunity has already been battling this thing for days, right? So you have macrophages, you have dendritic cells that have been doing their job, they've been eating these viruses, these bacteria, they've been digesting it. And so what they're going to want to do is that they can't do this forever, is they're going to want to recruit the B cells and T cells that can fight back more specifically and more effectively. And so what a class 2 MHC does is it's used by phagocytic cells like macrophages and dendritic cells. When they engulf that pathogen and break it down, just like the infected cell, they'll take pieces of the antigen, mount it on a class 2 MHC protein, and then display that on the outside. And that way, as a dendritic cell, let's say that the coronavirus gets in the back of your throat. Well, the first thing it's going to run into is not going to be a B cell or T cell, because you only have 2 of those in your whole body, right? So, but you have millions of dendritic cells and macrophages. So those macrophages are going to attack it, they're going to eat it, and then they display it using a class 2 MHC. And now what they're going to do is they just literally just move around your lymphatic vessels. They're going to search through your whole body to try to find those 2, 3, or 4 B cells and T cells that are specific for a coronavirus, because that's the only way that you're going to be able to mount a really specific attack. Okay, so that's the role of MHCs. And MHCs on their own are also really, really diverse. We have hundreds of different classes, not classes, but different types of MHCs, and they're actually so unique to each person that they can be kind of compared to like your fingerprint, because the MHC proteins that you make are directly related to what genes you have, right? To your unique genome. And so MHCs, while they do really good work here, can also become a problem later on, like if you need a heart transplant or any kind of organ transplant. We've talked about how it's doctors have to put a lot of effort into seeing if they have a good match. The reason why there might not be a good match and the reason why your body might reject it is because of these MHCs. If the MHCs are too different, they recognize them as being foreign, and then your immune system rejects it or even attacks it. Okay, so let's move on here. So with your acquired immunity, we're going to go through a kind of the different steps here, and then I'll kind of put it all together by looking at that comic, and I'll just move my face out of the way here. So the first thing we're going to look at here is the dendritic cell or the macrophages, okay, with the MHCs. They're going to display the pathogen after they engulf it, and that's the goal here is to trigger an adaptive immune response, which is to activate your B cells and T cells. Now, of course, this means there's going to be some delay in this response, because remember I told you that dendritic cell, that macrophage, is patrolling your body looking for those handfuls of T cells and B cells. So it's going to take some time to get there. But once it gets there, check out this picture. There's going to be this interaction that happens. Okay, and I don't really teach CD4 anymore, but since we have the time, I'll just kind of mention it. So here in this picture down below, let me see if I can zoom in a little bit. There we go. You have your macrophage that eats the bacteria, displays with the class 2 MHC. And here you have a T cell, this one's specifically called a helper T cell, we'll talk about that later, that has a receptor, like we talked about, right? Those 2 sticks with a variable region that matches that antigen receptor. And so you can see it's holding out that little green triangle. It says, hey, I know what that is. It binds to it. And then another molecule, another protein called CD4 will actually, it looks like it kind of reaches out and clamps on. It just strengthens that connection so that they can verify that it's the correct thing. And at that point, that's going to activate this T cell and trigger your active immune response. Okay. Now, since I mentioned it, T cells, the helper T cells, which is abbreviated TH, are really important because they are kind of the liaison between the innate immunity and the active immunity. What I mean by that is, remember, these T cells and B cells, they have receptors, right, for this antigen, for coronavirus. But because there's only like 2 of these and 2 of these in your entire body, the chances that they're going to bump into the coronavirus themselves is pretty low. And so what's going to happen is that the helper T cell, which you also only have a couple of, is also going to kind of spread out, and they're going to try to find a macrophage or a dendritic cell that's presenting with class 2 MHC. And once they do that, here's the really cool part. The helper T cell, this is where they get their name, can send out some chemicals like interleukin-2 and other cytokines. Cytokines are chemicals that can be used to activate the lymphocytes. And when those chemicals get released, they release them into your bloodstream, into your lymphatic vessels, and now from a long distance, they don't have to actually come in physical contact with each other. They're basically going to get a message, it's like a text alert that's saying, hey, the T cell, the cytotoxic T cell, the TC right here, and the B cell, you guys are needed, you guys should be activated, okay? And so helper T cells are really important for kind of spreading that message, and you'll see that as we look at things in the big picture. Speaking of big picture, here is how it works. Here's a diagram showing how the helper T cell can activate this process. Now, there are a lot of arrows going around everywhere, and that just shows that there's a lot of different ways to activate this process, right? We don't want to make it so strict and only create one pathway that there's only one way to activate your immune system. There's a lot of different ways you can activate it. So up top here, you have your antigen with what we call the primary or first exposure, or primary immune response. This means that your body's never seen this antigen before, and it's shown up in your body for the first time. And what we want to do, the end of the story here, is we want to activate these 3 right here. Okay, if we activate the B cell, the B cell can then talk to and activate the helper T cell and the cytotoxic T cell. If the cytotoxic T cell gets activated, that can stimulate and activate everybody else. And of course, the helper T cell can talk to everyone too. Okay? So whether it's the antigen directly comes in contact with the B cell and activates it, or if the antigen comes in contact with the infected cell that a cytotoxic T cell can recognize with MHC2, or if the helper T cell is activated by a macrophage, the use of cytokines and interleukins are going to stimulate and activate these lymphocytes, the B cells and the 2 helper T cells. Okay? And when that happens, we're going to see 2 kinds of responses. The humoral response, which specifically looks at what's happening with the B cells, and then the cell-mediated response, which is all about what's happening over here with the cytotoxic T cells. Okay? Again, note just that this can be activated all independently. So any of these 3 can be activated. They're not just waiting only for the helper T cell. Okay, but the helper T cell definitely helps. So let's take a look at the humoral response. Just time check here, 22 minutes. I'm trying to keep this kind of short because no one likes watching videos that are too long. But the humoral response, what we're looking at is what happens with those B cells, right? When a helper T cell activates the corresponding B cell, sorry, some of these points are out of order. I'll just give it to you all at once. The B cells are going to go into something called clonal selection. And that is not on this slide right now. I kind of wish right now that I put it on there. But if you want to write that word down, clonal selection means that 1 or 2 B cells, those 1 or 2 B cells are going to start to clone themselves. Okay, and you can see that in this picture over here on the right side. Here is a helper T cell that has come in contact with a infected cell, right? Oh, sorry. This is a macrophage that's presenting with MHC2. That helper T cell will activate and go through its own clonal selection, which means that it's going to clone itself many, many times. It's going to go from 2 cells to thousands of cells. And all those thousands of cells will then release, I'll zoom in a little here, those interleukin-2s, other cytokines, and then that'll activate the B cell. Now here's where clonal selection happens for B cells. When the B cell gets activated, it's going to, again, replicate hundreds of thousands of times, maybe even millions, and it'll split into 2 groups. Okay, now the 2 groups are what are known as your effector cells and your memory cells. The effector cells are going to have an effect for the battle right now. Those are the cells that are going to go into immediate action to take out whatever pathogen's infecting you. The memory cells are going to be set aside for later, and we'll come back to those at the very end. So when B cells go through clonal selection, they're going to differentiate into 2 types of cells, the memory cells and the effector cells. And for B cells, the effector cells have a special name. Those are called plasma cells. And these plasma cells will make those antibodies that I mentioned earlier. Right? You guys have probably heard about this before, like, oh, you need antibodies in order to fight back. Well, antibodies don't just show up out of nowhere. They have to be made by the specific B cell that is for that specific pathogen. In other words, if this is the B cell for coronavirus, then all these millions and millions of antibodies that are secreted are going to be made specifically with the same receptor that you see on the B cell that's going to be able to identify, bind to, and disable the coronavirus. Okay, and so the picture doesn't really show it, but imagine if you have millions of plasma cells throughout your body producing, each of them, millions of antibodies. The number of antibodies in your bloodstream is going to go up, and that's going to mount a really awesome attack to take out the pathogen. Now, what do these antibodies actually do? Though, there's a couple different actions that they can have. And this diagram here, if you're working on this in the book, is on page 913. It's figure 43.19. What they're going to do is a number of things. One is viral neutralization. This is kind of the easiest version. Antibodies, because they have binding sites that are specific to those epitopes, can basically bind to that virus and cluster around it, and now the virus is pretty much useless. It can't sneak into cells, it can't inject DNA into their host cells. It's basically being shut down. Another thing is some antibodies, they're called pentamers. So this one right here, you'll actually take 5 antibodies and link them up in a ring. And when all 5 of them grab onto a bacteria, that's going to cause something called agglutination, which causes the bacteria to basically clump up together. And that basically holds them and prevents them from spreading and moving them around. Similar idea with precipitation. If you have antigens that are soluble, that are, in other words, dissolved into water, the antibodies can actually grab onto a bunch of them, and when you pull things out of solution, that you get a precipitate, right? You get an actual solid that builds up. And why is that a good thing, to have solids or to have things clumped up or even disabled? Because what that means now is it's easier to do phagocytosis. So even though we're using the B cells and T cells now, it doesn't mean that the leukocytes we talked about earlier, right, your innate immunity, that they're just kind of sitting back and not working anymore. They are working side by side with these lymphocytes, and so now that everything is kind of clumped together, it's going to make it even easier for your macrophages and neutrophils and dendritic cells to locate the pathogen, engulf it, and carry out phagocytosis. Another thing that can happen, this is maybe a little too much for our class, but these antibodies can also activate the complement system and cause pore formation. This is really for cells that have been infected. So if these antibodies see, let's say, an MHC1 molecule presenting the antigen, the antibodies can actually recruit these proteins that will arrive at the surface of the membrane, insert themselves into the cell membrane, and then create a pore that basically leads to cell lysis. In other words, it causes the cell to fall apart. And that's just one way to trigger apoptosis. So that is what the humoral response is all about. Okay. Now, again, the thing you want to kind of imagine here, the thing you want to keep in mind, is that when something takes over in your body, there's really 2 places where they can be found, right? If you have coronavirus, even if you don't show the symptoms yet, they can be found in 2 places. One is inside your cells, right? If the virus has infected the cells inside your lungs, they're hiding inside your cells. The antibodies aren't really going to be able to get there, okay? And that's going to be the job of the cytotoxic T cells. But any viruses that are floating around out in the open, in your interstitial fluid, in your lymphatic fluid, in your bloodstream, anywhere it's just free floating in fluid, antibodies can get there and they can shut them down before they have a chance to infect other cells. Okay, so let's move on to the cell-mediated response. We're almost at the end here. In the cell-mediated response, let me move my face over here. We're looking at the right side of this table, okay, of this graphic. And I know it's a little small right now, but you've got this in your book. In the cell-mediated response, we are going to be recruiting these cytotoxic T cells. And cytotoxic T cells are really, really, really effective because they secrete proteins that trigger apoptosis in infected cells. And so they, kind of like natural killer cells, can kill infected cells, but it's much, much more specific. Now, the way they do that is, we'll actually start with this picture down here, is when cytotoxic T cells come in contact with an infected cell. Remember, infected cells are using class 1 MHC molecules, right? So they're displaying the antigen there. And the receptor protein here recognizes it, they're going to use a different protein. It's similar to CD4. This one's called CD8. That's just unique to cytotoxic T cells. And they're going to bind to it and basically say, hey, I see you're infected. I'm sorry, it's time to take you out. And at that point, two things get released. You get perforin and you get granzymes. What perforin does is it gets its name from making perforations, or pokes little holes. What it's going to do is it's going to poke holes in the whole the membrane of the infected cell. And then what it does is it releases granzymes, which are enzymes that initiate and trigger apoptosis, and that's going to see, as you can see in the picture down here, that's going to cause that cell to basically self-destruct and kill any pathogens inside along with it. Now, I kind of forgot something here, which is that, like the B cells, the cytotoxic T cells have to go through clonal selection as well, right? You don't want just two cytotoxic T cells for coronavirus trying to do all the fighting for the whole body. So like the B cells and like the helper T cells, they're going to go through clonal selection. They're going to divide millions and millions of times, and now your body is filled with these cytotoxic T cells, and they're going to go and do the fight. At the same time, though, just like the B cells, when they go through clonal selection, you're going to see two groups being made. You're going to have your active cytotoxic T cells, which are going to do everything that we looked at here, but you're also going to have your memory cells. And those cells are going to kind of sit in the background. Their job is to help build secondary, they're going to build your immunity for a secondary infection, which we'll look at later. Okay, so it's kind of a brilliant design here, because what you're doing is you're taking care of the infection now, but you're also assuming that if the pathogen, if this virus is around now, it's probably going to be around later, and you're already preparing for that in the future by making memory cells, and that's going to be memory B cells, memory helper T cells, and memory cytotoxic T cells. So that is the cell-mediated response. So here's kind of your big picture, and as we've talked about, you've got your humoral and cell-mediated response. There's a lot going on in this picture, and unfortunately the way the book is made is that it kind of jumps around. Like it's got some really good pictures here where it shows you the big picture of who's activating who and how clonal selection gives rise to different effector cells and memory cells. What I don't like about it, though, let me move this out of the way, is that it leaves out a lot of the details that we saw in previous pictures, right? Like the ones that we see here with the MHC2s and with the B cells interacting with class 1 MHCs. And that's why, going back to my comic that I made, if I pull this up, at the very, very end on the last page, okay, you can read through all the cheesy comic stuff. What I've done is basically I've combined all the diagrams of the book onto one page for you. And you can print this out. This is a great study guide, but it looks exactly the same as the one that you see here, right? But the nice thing is that there are the little details of the class 2 MHCs and the cytokines and all that. I did leave off the CD4 and CD8. It's a little beyond the scope of our class. By the way, I was talking to one of my students who's home right now because of coronavirus, and he's a Mills graduate, and he's in med school right now taking an immunology class, and they're learning all the same stuff. He's like, this is really easy because I already learned this back in AP Bio. So always a good affirmation of what we're doing here, even if it's a little bit more than you need to know. I think it's manageable. You guys are smart. Okay, let's land this plane. It's already 32 minutes in. Okay, so quick thing, memory cells. We've already talked about this, but memory cells remain even after the infection. So once you've fought off the infection, all the active T cells, the active B cells, those plasma cells, and the active helper T cells, they actually done their job, they die off, they go away. But the memory cells, they're going to stick around and help with a secondary exposure. And this graph, which is really commonly seen, and you're probably going to see this with stuff that comes, after the coronavirus stuff passes. This shows what your body's doing in terms of its antibodies and its response. Here, this first little hump, that's your response to your primary immune response, right? This is the first time that you are exposed to it, and that's going to cause you to have active immunity, because when you get infected the first time, you're going to build up all those memory cells and you're going to build up all those antibodies, so that the second time you're exposed to it, the response is going to be much quicker and it's going to be a lot more heightened. You can see the number of antibodies is going to be much higher than the first one. And that's basically the reason why they say with most diseases, if you get it once, you're not going to get it again, right? But if you get a different version of it, so like the flu influenza virus mutates quite a bit, And so every year the flu virus is different. Well, that means that your body's always trying to play catch up, which means the first time you're exposed to it, you're going to have to go back through the active immunity process and activate those B cells and T cells all over again. So this really only works, this active immunity only works the second time if you've already been exposed to it. Now, there's another way to kind of trick the body into getting this immunity without actually having to be sick, and that's called passive immunity. And in passive immunity, you are going to artificially inject antibodies into a non-immune person. And I forgot to mention, this is kind of different from vaccinations. Okay, technically vaccinations are a form of active immunity, because what you're doing when they come up with the coronavirus vaccine one day, they're working on it, is they're going to inject parts of the antigen or a non-pathogenic form. In other words, one that's not going to get you sick. They're going to take those, they're going to modify it, and they inject it into the person, and the body doesn't know that you're getting sick or not. So it's gonna make the B cells and T cells and antibodies and all of that, and it makes the memory cells without you actually having to get sick. And that's the value of vaccinations. Okay, and obviously, there are some folks that are kind of anti-vaxxers, and we just got to be patient with them and educate them on the science behind this. Now, passive immunity is a little bit different. That means that you're actually injecting or giving antibodies directly to a non-immune person, and that's going to provide immediate short-term protection. This is sometimes used with people that are immunocompromised, that don't have all those cells they need to activate everything we just talked about. What we can do is just give them antibodies for the short term, so that any viruses or bacteria that are floating around can be labeled and clumped together like we talked about, and it gives their innate immunity a chance to kind of catch up. Another really cute and sweet way to get passive immunity, and this is really relevant to my family right now, is you can get natural immunity from the mother via the placenta or the milk. So right now, my wife, I was kind of joking with her last night. She wasn't too thrilled about it, but coronavirus is kind of all over the place, right? And I gotta close this up because we have a doctor's appointment for the baby. But whatever my wife comes in contact with, her body's going to go through that immune response. It's going to build up antibodies, and the really cool thing is some of those antibodies can actually cross over through the placenta, so that my son is actually getting those antibodies before he's even born. And so even though he's born with a clean slate with a brand new immune system, he's actually mom's going to give him a little preview of what he might run into. And also through breastfeeding, she can also pass on antibodies through her milk. Okay, I'm going to let you read up on the immune system and transplants and transfusions on your own. We talked about type A, B, and O blood, so this is just all kind of related to the immune response and foreign antigens. And then the book does a pretty good job of covering immune system disorders. You've heard of autoimmune diseases. A lot of times what that basically means is that your immune system, for some reason, thinks that a part of your body is an enemy and it attacks it. So if it's your joints, it's going to cause arthritis. If it's I can't remember exactly right now, but I was just reading about it. If it thinks it's your histones or your DNA, then you're going to have lupus. And if it's the myelin sheath around your neurons, it's going to be multiple sclerosis. So there's a lot of different disorders that are classified as autoimmune diseases, and they all work the same way, where your T cells and B cells, for some reason, recognize the MHCs or certain proteins as enemies, and they attack it. And we've also talked briefly about allergies, where you have these antigens that really aren't harmless, right? Pollen and peanut oil, not really harmless, but your body overreacts to it, and those mast cells will release huge amounts of histamine, then that trigger an allergic reaction. And that's why we take antihistamines, right, is to bring down that vasodilation and lower the symptoms. But in some cases, like if you have an anaphylactic shock, your body's releasing so much histamine that it actually dilates all your blood vessels at the same time. Your blood pressure actually can drop, and that can be a really dangerous thing. So that's something that people that work in the ER and hospitals are always looking out for. Okay, I'm gonna have you read up on AIDS because I gotta go, but I think that's basically, it's pretty self-explanatory. AIDS will lead to you being immunocompromised. All right, I got to roll to this appointment. I hope that's helpful. If you have any questions about anything, if I went too fast or I wasn't clear, or maybe I made a mistake, I do that all the time, just leave a comment below and I will address it when I get back to you guys. Tonight, you guys are working on the reading guide for this chapter. And then tomorrow, on Thursday, we're going to have our first live stream. So for both second and third period, that's going to be at 9:30. We're going to be online here, and I can answer any questions you have. It's a chance for us to check in, ask questions. I wish I could see all of you guys, but at least you guys get to see me and make me look stupid. So hope you guys are doing good out there. If you haven't already watched my intro video that I made yesterday or 2 days ago, do watch it. We talk a little bit about what it means with all this shelter-in-place stuff and why it's so important to stay isolated. All right, you guys stay safe out there. Take care. If you have any questions, let me know. Have a good one. Thanks.